So imagine a rotting spruce log that's just buried deep under this freezing alpine snowpack.
Right, in an environment that is basically hostile to almost everything.
Exactly. I mean, the wood is totally saturated with highly acidic resins, toxic heavy metals, and the ambient temperature is hovering just barely above freezing.
Which means any water inside a living cell is like mere degrees away from forming these microscopic ice daggers.
Yeah, ice daggers that would literally shred the cell membrane from the inside out. And if that's not bad enough, the immediate area is heavily populated by this other organism that's just pumping out lethal, liver-destroying neurotoxins.
It is a literal death trap.
It really is. To almost every living thing on planet Earth, this environment is a nightmare. But to this one tiny, unassuming brown mushroom called Cuneromices lignicola, which is commonly known as the conifer woodcuft, this whole toxic, freezing setup is basically an all-you-can-eat buffet.
It's wild. It's a phenomenal organism to study.
So that is our mission for today's deep dive. We are taking this massive stack of ecological studies, taxonomic records, and these super advanced chemical profiling papers, and we're going to extract the secrets of a fungus that essentially acts as a biochemical survivalist.
I am so ready for this.
Yeah.
Because, to the untrained eye, or even to like a super experienced hiker out for an early spring walk in a subalpine forest, this thing just looks like another little brown mushroom.
Right.
Mycologists actually have a running joke about this. They call them LBMs.
LBMs, like literally little brown mushrooms.
Yeah, exactly, because you can walk past 10,000 of them and they all look virtually identical. You would never guess that beneath that totally mundane exterior is an organism that's constantly engaged in this extreme chemical warfare and like complex biological engineering.
It's basically an undercover operative. You look at the setting, these montane and subalpine forests across the northern hemisphere, and it's late spring. The rest of the forest is practically dormant, you know.
Absolutely. The soil is frozen hard.
Yeah, the deciduous trees haven't leafed out yet, and they're these massive, harsh, melting snowpacks dominating the whole landscape. And pushing right up through that icy duff, just shivering at the edge of the freezing runoff, is the wood tuft. It thrives where others instantly perish.
It really does.
But looking through the historical sources you brought in, it seems like its ability to blend into the background was almost too good. Like the scientific community spent over a century just trying to figure out what to even call it.
Oh, the history of its name is a fascinating window into the history of mycology itself.
Oh.
I mean, it took roughly 150 years of scientific argument to finally pin down its identity.
150 years.
Yeah, and that wasn't due to scientists being careless or anything. It was because the wood tuft is this morphological shapeshifter. Its physical appearance changes so drastically based on its environment that early scientists were just constantly fooled into thinking they were looking at completely different species.
Okay, so let's wind the clock back to trace that confusion. The timeline starts in 1872 with an American mycologist named Charles Horton Peck, right?
That's right.
He encounters this little brown mushroom in the woods, examines it, and names it Agaricus lignicola. Now, my understanding of the genus Agaricus today is that it includes, like, the common white button mushrooms you buy at the grocery store.
Yeah, the pizza mushrooms.
Right, the pizza mushrooms. So our freezing, wood-eating survivalist doesn't seem to belong anywhere near a grocery store button mushroom. Why did Peck put it in that group?
Well, we really have to put ourselves in the shoes of a 19th century botanist here. In 1872, your laboratory equipment was essentially, a sharp knife, a basic hand lens, and your own senses.
I know DNA sequencing.
Exactly. DNA sequencing or even advanced microscopy was decades and decades away. Consequently, the classification system was incredibly crude. The genus Agaricus became this massive, chaotic dumping ground.
Just a catch-all for whatever they found.
Right. In the scientific literature, we call this a polyphyletic grouping.
Poly, meaning many, and phyletic, referring to evolutionary branches or lineages. So basically a grouping that contains a bunch of totally unrelated branches.
Exactly that. It wasn't a true evolutionary family tree at all. It was a visual sorting bin. If you found a mushroom that had a stem, a cap, and fleshy gills underneath that produced spores, the default action was just to toss it into the Agaricus bucket.
Just file it away and deal with it later?
Pretty much.
Yeah.
They simply didn't have the tools to differentiate the subtle microscopic traits that actually define true evolutionary relationships.
I can just imagine the filing cabinets of 19th century universities totally overflowing with thousands of entirely unrelated mushrooms all labeled agaricus.
Oh, it was a mess.
So as time went on and microscopes obviously improved, they had to start sorting that lost and found bin. And our mushroom gets its cover blown for the first time. It gets moved into the genus Pholiota, becoming Pholiota lignicola.
Yes, but the taxonomic musical chairs definitely didn't stop there.
Right. Looking at the records, later it's called Pholiota vernalis. Then someone suggests Caenoromyces vernalis. It's basically bouncing around using aliases for decades.
The bouncing around happened because scientists started using macroscopic features to subdivide that massive agaricus bin.
Hmm.
They started looking at the color of the spores when they dropped in a mass, or how the gills physically attached to the stem.
So getting a little more specific.
Right, so the genus Pholiota was created from mushrooms that typically grow on wood, produce brown spores, and usually have very distinct scaly or slimy caps.
And our subject grows on wood and drops brown spores, so it seems like a good fit.
It seemed like a fit, yeah, but it completely lacks those dramatic scales on its cap. It's remarkably smooth.
So it was kind of a square peg in a round hole. It had some traits of a Pholiota, but not enough to make the mycological community actually happy.
Which led to endless debates.
Mm-hmm.
I mean, it wasn't until 1984, over a century after Peck first found it, that a mycologist named Scott Redhead finally ended the argument.
Wow, 1984. That's practically yesterday in terms of biological history.
It really is. He cemented its identity and placed it definitively in the genus Caenoromyces, officially establishing the name Caenoromyces lignicola.
And the sources mention that Redhead didn't just look at the mushroom with a hand lens. He looked specifically at the spores under high-powered magnification. The defining piece of evidence was something called a truncated apical germ pore.
Yes, a very specific microscopic feature.
So let's break that down for someone who hasn't peered through an electron microscope recently. I know spores are effectively the microscopic seeds of the fungus. What exactly is a germ pore?
Well, think of the spore as a microscopic, heavily armored egg. Its job is to survive the journey through the air and just sit in the soil or on wood until conditions are absolutely perfect.
Okay, makes sense.
But when conditions finally are perfect, the organism inside have to get out. The germ pore is a specialized, thinned-out region on the spore's thick outer wall. It's essentially an escape hatch.
Oh, I see.
Right, so when moisture and temperature trigger germination, the internal pressure of the spore builds up immensely, and the new fungal thread, the hyphae, pushes out through that weakened pore to start a whole new mycelial network.
And Redhead noted this pore was apical and truncated. So apical just means it's located at the very tip or apex of the spore, right? Like the top of the egg.
Yes, exactly.
But truncated means flattened or cut off rather than perfectly round. So how large of a structure are we actually talking about here?
The measurements in the literature show this pore is roughly 0.5 to 1.0 micrometers across.
1/2 millionth of a meter. That is wild. A microscopic flattened escape hatch on a spore was the definitive piece of evidence that moved an entire species into a totally different taxonomic genus.
It really highlights how incredibly precise modern mycology has become. That specific pore structure, combined with the fact that its spores are entirely smooth, unlike a lot of its relatives, and that its stem is non-scaly, just aligned it perfectly with the genus Caenoromyces.
So Redhead's work was basically a triumph of microscopic taxonomy.
Yep.
But while the microscopic details are what finally named it definitively, the macroscopic details, what you actually see walking through the forest, are what caused all the confusion in the first place. You mentioned earlier that it's a shapeshifter.
Oh, completely.
The cap is small, usually just 1 to 4 centimeters across, but the literature consistently uses this specific word to describe it.
Hygrophanous.
Hygrophanous is such a brilliant botanical concept. It describes tissue that physically changes color depending on its moisture content.
So it's not a chemical reaction or something?
No, not at all. It is not a chemical reaction or a pigment change. It is entirely optical.
Oh, like getting out of a swimming pool in a light-colored t-shirt.
Yes.
The wet fabric looks like 5 shades darker than the dry fabric because the water trap between the fibers changes how the light reflects and passes through it.
That is a flawless analogy. The cellular structure of the wood tuft's cap acts exactly like a sponge. When it is fully saturated, which happens constantly in its melting snowbank habitat, the water fills the microscopic spaces between the cells. So the light interacts with it differently.
Exactly. Light passes through it differently, making the mushroom appear this rich, dark, cinnamon brown, almost translucent, kind of like wet glass.
Wow, okay.
But as the ambient air hits the mushroom, the water evaporates. And the crucial detail that fooled so many early mycologists is how it dries. It doesn't dry uniformly.
Right. The sources say it dries from the center, radiating outward.
Yes, the very center, the apex of the cap, loses moisture first. So that specific spot suddenly fades to a pale yellowish tan or even a stark white.
While the rest is still dark.
Right, the outer rim of the cap is still holding water, so it remains that dark cinnamon brown. It creates a really striking two-toned bullseye effect.
I totally see the problem now. If Charles Horton Peck finds it at 8:00 a.m. while it's covered in freezing morning dew, it's a solid dark cinnamon color.
Yep.
But if another botanist finds the exact same mushroom at 2:00 p.m. after a dry spring breeze has hit it, it looks like a completely different two-tone species.
And if they find it a day later when it's completely dry, it's entirely pale yellow.
You can easily see why it generated a century of aliases. And it isn't just the cap playing these visual tricks, right? The stem or the stipe has its own distinct characteristics. The literature describes it as becoming vinaceous brown.
Vinaceous meaning wine-colored, so a deep purplish red-brown.
Okay, but the key identifier is the progression of that color, isn't it?
Correct. The dark coloration originates at the very base of the stem, right where it actually attaches to the rotting wood, and it slowly creeps upward toward the cap as the mushroom matures.
So it looks like it's blushing from the bottom up.
That's a good way to picture it. And this dark, smooth stem heavily contrasts with another feature mentioned in the taxonomy, which is an evanescent fibrillose membranous veil.
Evanescent meaning vanishing or fleeting.
Right. When the mushroom is very young and still actively pushing through the wood, it has this delicate cobweb-like membrane stretching from the stem to the outer edge of the cap.
What's the purpose of that web?
Its purpose is to protect the developing, really fragile gills from dry air or predatory insects. But as the cap expands, like an umbrella opening up, this thin membrane tears and usually vanishes completely.
So it's not a permanent feature.
No. If you were really lucky, you might just see a faint, dark ring high up on the stem where a few falling spores got trapped on the tiny remnants of the web.
So the observer out in the field is dealing with a cap that changes color by the hour, a stem that darkens over days, and a protective veil that just disappears entirely. It really is a master of disguise.
It makes field identification incredibly challenging.
And the plot thickens even more when you look at the geographic data because we also have distinct regional variations. The taxonomy notes mention Cuneromyces lignicolor conicus.
Yes, the conicus variety.
This specific variety was originally documented in Idaho and then randomly appeared in collections from Austria. Why isn't it just considered a standard wood tuft? Like, what makes the conicus variety distinct?
Well, the conicus variety provides a really wonderful example of microevolution and geographic isolation. While the standard lignicolous starts out slightly rounded and eventually flattens out almost like a pancake as it ages, the conicus variety maintains a sharply umbonate cap.
Umbonate. That means a distinct raised bump in the exact center of the cap, right? Almost like the boss on a Viking shield.
Or a tiny, prominent wizard's hat.
Ah, yes, a wizard's hat.
So it has a very distinct conical shape, hence the name conicus. But the true differentiator, once again, requires a microscope. The conicus variety has consistently capitate Tylocystidia.
We are definitely going to need to define Tylocystidia for you all listening.
Of course, Tylocystidia are microscopic, sterile cells that line the very edge of the gills. They don't produce spores themselves. Their function is still somewhat debated in mycology, but we generally believe they act as microscopic bumpers.
Bumpers, like on a car.
Kind of, yeah.
Yeah.
They keep the adjacent gills spaced slightly apart so that when the spores mature, they can drop straight down to the wind currents without getting stuck to the neighboring gill face.
Oh, that makes perfect sense. And capitate describes the shape of these bumpers.
Yes. Capitate means having a distinct, swollen head at the top, very much like a bowling pin.
Okay, got it.
In the standard wood tuft, these sterile cells are highly variable. Some are lobed, some are irregular, some are thin. But in the conicus variety found in Idaho and Austria, every single one of these cells is shaped perfectly like a little bowling pin.
That is an incredibly minute detail.
It is, but it proves that this organism has distinct genetic populations operating completely globally.
That attention to microscopic detail is just staggering. And I think it transitions perfectly into the actual day-to-day life of this fungus.
Yeah.
Because that hygrophonous nature we talked about, the fact that its entire body acts like a water-absorbent sponge, is actually the biggest clue to its overall survival strategy.
It absolutely is.
The moisture that causes it to change color is the exact same resource it leverages to completely dominate a very specific, totally hostile environment. The studies refer to this as the snowbank strategy.
This is where the ecology of the conifer woodtuft becomes truly exceptional. It essentially breaks the standard rules of fungal foraging.
Yeah, because when most people think of mushrooms, they think of autumn. Warm, damp soil, falling leaves, the smell of decay in October. In fact, a close relative of our subject, Cuneramyces mutabilis, the summer woodtuft, does exactly this.
Right. It waits for the easy, temperate days of summer and fall.
But our Lignicula looks at a freezing, melting pile of ice in late spring and decides, yep, that is the optimal time to emerge. What evolutionary pressure drives an organism to choose a freezing snowbank over a nice, warm autumn afternoon?
The driving pressure is the avoidance of competition. Pure and simple.
Really, just avoiding the crowds.
Exactly. The forest floor in late summer and autumn is an absolute microbial war zone. I mean, millions of spores from thousands of different fungal species are raining down on every single available surface.
So it's incredibly crowded.
Highly crowded. If a tree falls in September, it is immediately colonized by incredibly aggressive, fast-growing fungi. Those autumnal species are thermophilic, meaning they love the warmth and require it to metabolize quickly.
So if our little brown mushroom tries to fruit in September, it just gets outcompeted, overgrown, and basically starved out.
Without a doubt. So, to survive, it adopted a strategy of extreme phenology.
Phenology being the study of cyclic and seasonal natural phenomena in relation to climate.
Right. By adapting its entire physiology to function at near-freezing temperatures, it can colonize and consume its chosen food source, while all the thermophilic warm-weather fungi are still totally dormant, just waiting for summer.
Wow.
It relies on the slow, steady drip of near-freezing water from melting subalpine snowpacks. It will literally fruit underneath the snow crust, or just millimeters from the melting ice, completely monopolizing the rotting wood before the summer competitors even wake up.
Arriving early to beat the crowd makes perfect sense for real estate. But this brings up a massive biological paradox that the sources highlight. It arrives early, but it is incredibly picky about what it eats. It only consumes conifer wood. Specifically, the literature notes it grows on spruce, the Picea genus, and fir, the Abies genus.
Yes, very specific host trees.
And anyone who has done woodwork or built a campfire knows that conifer wood is packed with sap, pitch, and resins. It is notoriously toxic and highly resistant to rot. Why on earth would a fungus adapt to eat the hardest, most toxic food source on the coldest day of the year?
You have identified the core genius of its ecological niche.
Like,
You're right, conifer wood is highly recalcitrant. Structurally, it is packed with complex lignin.
lignin.
Think of it this way. If the cellulose in wood is the steel rebar of a building, the lignin is the hardened concrete poured over it. It is an incredibly dense three-dimensional polymer.
Okay, so it's structurally super tough.
Very tough. But more importantly, the wood is loaded with acidic resins, phenolics, and tannins. Trees evolved these chemical compounds for one specific reason: biological warfare.
To fight off infections.
Exactly. They are chemical deterrents designed specifically to poison bacteria and fungi, preventing the tree from rotting while it's alive.
So the tree is actively defending its carbon even long after it dies. Conifer wood is basically a hostile, acidic fortress. So how does the wood tuft reach the walls?
It mounts a highly specialized enzymatic counterattack. Fungi that decay wood generally fall into two broad categories: brown rot and white rot.
I've heard those terms used by arborists.
Right, so brown rot fungi only have the chemical tools to eat the cellulose, the rebar. They leave the tough lignin concrete behind, which is why rotting pine logs often look like crumbly brown cubical blocks.
Oh, I've totally seen that in the woods, those little brown cubes of wood that crumble in your hand.
Exactly. The wood tuft, however, is a white rot fungus. It possesses the heavy artillery required to completely obliterate the lignin itself, leaving behind a stringy, bleached white residue.
So what exactly is the heavy artillery here? How do you biochemically break down organic concrete?
It wields powerful oxidative enzymes called lactases. Researchers have specifically isolated, purified, and studied the lactase enzyme from the Cuneramyces genus, and they named it Kimlac.
Kimlac.
Yes. This enzyme is a biochemical marvel. It uses copper atoms at its core to physically strip electrons away from the lignin polymer, causing the entire rigid structure to just destabilize and fall apart.
But doing that in a clean, temperature-controlled laboratory is one thing. Doing it inside a freezing, highly acidic, rotting spruce log is a completely different story. Most enzymes are incredibly fragile.
Extremely fragile.
If the pH level, the acidity is too high or too low, the enzyme denatures. It basically unfolds and becomes totally useless. So how does Kimlek survive the acidic resins of the spruce tree?
Here's the amazing part. It doesn't just survive the acidity, it actually requires it.
Wait, really?
Really. Kimlek is perfectly tailored for its toxic environment. The decaying conifer logs are highly acidic due to all those defensive tannins and resins we talked about. The researchers found that Kimlek has an optimal activity at a highly acidic pH of around 4.0.
Oh, wow.
It thrives in the exact harsh conditions that would instantly destroy the digestive enzymes of competing fungi.
That is staggering. It essentially uses the tree's own chemical defense mechanism as its optimal operating environment. It turns the tree's shield into its own weapon.
It is a beautiful example of evolutionary adaptation. But the fungus doesn't just indiscriminately blast the wood with enzymes. It plays a highly strategic waiting game based on elemental chemistry. It constantly monitors the carbon-to-nitrogen ratio, the CN ratio of the dead tree.
Well, fungi need carbon for energy, but they need nitrogen to build proteins and enzymes, right? And when a tree first falls, I assume it's almost entirely carbon. The wood is just dense sugar and lignin.
Exactly. When the tree falls, the carbon is overwhelmingly high, and the nitrogen is way too scarce to support massive fungal growth. So Ecological just waits patiently as a dormant mycelial network within the wood.
Just biding its time.
Right. And over time, as weather and slow bacterial action begin to alter the wood, and as nitrogen slowly accumulates from the surrounding environment, that CN ratio begins to shift.
And when it hits the sweet spot,
The moment the ratio hits a favorable balance, the fungus rapidly ramps up production of its chem-like enzymes, obliterates the lignin, and completely dominates the log before any other species can even mount an attack.
it is a literal masterclass in timing. But we have to address the physical reality of its environment again. It's secreting these enzymes into near-freezing water. To deploy this whole strategy, the fungus itself has to survive temperatures that normally destroy cellular life.
Yes, the freezing problem.
If a living cell freezes, the water inside it forms jagged ice crystals. Those crystals expand and literally puncture the cell walls and membranes, turning the tissue to absolute mush. So how does the wood tuft avoid freezing to death while just sitting in a melting snowbank?
Answering that question required scientists to move beyond microscopes and look at the molecular fingerprint of the organism. They utilized a technique called NMR metabolomics.
Okay, the sources mention proton nuclear magnetic resonance, or 1H NMR spectroscopy. From what I understand, this is essentially putting a tiny piece of the mushroom into a highly advanced MRI machine.
That's a great way to visualize it.
But instead of generating an image of tissues, the powerful magnetic field interacts with the hydrogen protons in the sample. And by measuring how those protons spin and resonate, scientists can draw a highly precise graph that identifies every single molecule present. It gives you the complete chemical inventory of the organism.
That is an excellent summary. It reveals the metabolome, the complete set of small molecule chemicals or metabolites within the tissue.
Right.
When they ran K. lignicula through the NMR spectrometer, they isolated 27 major metabolites. They found a complex soup of primary metabolites, which are the basic building blocks necessary for life, including abundant amino acids like glutamate and leucine.
But the real breakthrough discovery in the NMR data, the key to its snowbank survival, was a massive concentration of a specific sugar, a disaccharide called alpha-trehalose.
Trehalose, yes, it's a legendary molecule in the biological world, famous for conferring extreme stress resistance in organisms.
You know, I live in a climate that gets really harsh winters. And every November, before the deep freeze hits, I have to pour liquid antifreeze into my car's radiator.
A necessary chore.
Yeah, because if I just leave pure water in there, the water freezes, expands, and exerts enough hydraulic pressure to literally crack the solid metal engine block. The antifreeze lowers the freezing point and prevents that crystallization.
Is trehalose basically acting as a biological antifreeze for the mushroom?
It is acting exactly like antifreeze, but the physics of how it works at a cellular level are even more fascinating than what happens in your car. When the ambient temperature drops toward freezing, The wood tuft actively produces massive amounts of this trichlose.
Okay.
And these sugar molecules pack tightly against the delicate lipid membranes and internal proteins of the cell. They physically displace the water molecules.
So they push the water out of the way before it has a chance to turn into internal ice daggers.
Yes. And as it gets colder, the trichlose forms a highly viscous, glass-like state. It's a process called vitrification.
Vitrification, like turning into glass. Exactly. It acts as a biological scaffolding, essentially freezing the internal cellular machinery in place without any damaging ice crystals ever forming. The proteins are just locked in a protective glassy matrix until the temperature rises again.
And when it warms up?
The cell just resumes normal function like nothing ever happened.
That is incredible. And the NMR data also revealed something incredibly strategic about how the fungus deploys this antifreeze, didn't it? It wasn't spread evenly throughout the mushroom.
No, it wasn't.
The concentrations were significantly higher in the stem than in the cap. If you have a limited supply of antifreeze, why pump it all into the stem and leave the cap relatively unprotected?
Well, you really have to visualize the physical architecture of the mushroom in its specific environment. The cap might be pushed up above the snow, exposed to the slightly warmer, sunlit spring air.
Right, catching some rays.
But the stipe is the crucial structural foundation. It is buried deep within the freezing ice and snowpack, connected the cap all the way down into the rotting log. The stipe is the nutrient highway.
Ah, I see.
All the water and broken down lignin must travel up the stem to fuel the development of the spores in the cap.
So if the stem freezes and ruptures, the highway collapses and the mushroom dies instantly, regardless of how warm and safe the cap is.
Exactly. The fungus recognizes its structural vulnerability. It aggressively shunts its trichlose production down into the stipe to heavily armor its most exposed critical infrastructure.
Yeah.
It is highly localized, perfectly optimized chemical engineering.
It's brilliantly adaptive. But, the chemical profiling from the NMR didn't just reveal a clever antifreeze system. It exposed a much darker, highly toxic trait. The wood tuft is a hoarder.
Oh yes, it certainly is.
And the specific things it hoards are heavy metal.
This specific characteristic makes the conifer wood tuft both ecologically indispensable and a serious potential hazard. The metabolic profiling demonstrated that this species actively absorbs, transports, and concentrates extreme levels of heavy metals from its environment, specifically cadmium and mercury.
Cadmium and mercury are universally recognized as incredibly toxic to almost all forms of biological life. I mean, they bind to proteins, disrupt DNA, and shut down internal organs.
They are lethal.
So why would a living organism actively draw poison into its own tissue? And more confusingly, how does it survive carrying lethal doses of cadmium?
Well, fungi serve as the great subterranean recyclers of the forest ecosystem. Their vast mycelial networks are incredibly efficient at pulling necessary trace minerals like copper for those lipase enzymes we talked about, or zinc and iron out of the soil and wood.
They act like a giant sponge for minerals.
Right. However, the chemical transport mechanisms aren't always perfectly selective. Sometimes, as they forage, they pull up highly toxic heavy metals right alongside the essential nutrients.
There's accidental bycatch.
Basically. But to prevent these heavy metals from destroying its own cellular machinery, Ecological has evolved an ingenious internal vault system.
The sources refer to this vault as the cell wall fraction, or the $5. If a cell absorbs cadmium, the metal naturally wants to float around in the cytoplasm where all the vital enzymes and DNA are located.
Which would be disastrous.
It's like having a loose radioactive material floating around in a crowded room. I assume the vault system is a way of containing it.
That is exactly its function. The metabolic analysis showed that the heavy metals, particularly cadmium, are actively transported up the stem and into the mushroom's caps, but they are not allowed to remain in the cytoplasm.
So where do they go?
The fungus actively binds up to 95% of the accumulated cadmium directly into the complex polymers of its cell wall, the rigid outer shell of the cell, which is made of chitin and glycoproteins.
So it's literally building the poison into the physical bricks of its own walls, keeping the toxic elements permanently locked away from its vital internal organs.
Precisely.
It's the cellular equivalent of encasing nuclear waste in a solid lead box.
That's a great analogy. By trapping the cadmium in the structural wall, the heavy metal is completely immobilized. It cannot interact with or damage the metabolic engines of the fungus.
Which is an absolute feat of biochemistry. But it raises a huge red flag for anyone walking through the woods. This makes the fungus an incredibly efficient environmental filter. It is actively pulling mercury and cadmium out of the decaying wood and sequestering it in its caps.
Yes.
But it also means that if a human forager harvests this mushroom from an industrially polluted area or downwind from an old mining smelter, they are essentially eating a concentrated capsule of toxic waste.
It really underscores the golden rule of wild foraging.
Hmm.
You are never just eating a mushroom. You are eating a concentrated biological record of the environment it grew in. Even if a mushroom species is technically classified as edible, its metabolism will totally mirror the pollution of its habitat.
That's a chilling thought. And as it turns out, heavy metal poisoning from industrial runoff isn't even the most terrifying danger associated with the conifer wood tuft.
Not even close.
Despite its rugged survival skills, its biological antifreeze, and its acid-secreting weaponry, this mushroom is constantly stalked by two lethal threats in the wild. One is a threat to the humans who try to pick it, and the other is a really bizarre threat to the mushroom itself. We have to talk about the deadly doppelganger.
This is where the narrative shifts from fascinating biochemical adaptations to a stark, critical public health warning.
The sources introduce a species called Galerina marginata, widely and very appropriately known as the deadly galerina. And this isn't just a mushroom that looks somewhat similar if you squint at it from a distance.
No, not at all.
It grows on the exact same conifer logs at the exact same time of year, literally sharing the same square inch of wood as our wood tuft.
They occupy the exact same ecological niche.
Yeah.
To the naked eye, a cluster of Kunoromyces lignicola and a cluster of Galerina marginata can be virtually indistinguishable.
That is terrifying.
They are both small, brown, hygrophonous mushrooms growing in dense clusters on dead wood. But the chemical difference between the two is quite literally a matter of life and death.
Because the deadly galerina is packed with amatoxins. For the listener who might not be totally familiar with fungal toxicology, we need to explain just how catastrophic amatoxins actually are. This is the exact same lethal compound found in the infamous death cap mushroom, Amanita phalloides.
Yes, the heavy hitter of mushroom toxins.
What exactly happens inside the human body when amatoxin is ingested?
Amatoxins are universally considered some of the most insidious and effective naturally occurring poisons on the planet. Structurally, they are bicyclic octapeptides.
Meaning their molecular structure is a tight double ring of 8 amino acids.
Right. And this double ring structure makes them incredibly rigid and stable. You cannot neutralize them with heat. Boiling the mushroom, frying it, or freezing it does absolutely nothing to the toxin.
So cooking the mushroom in a stew just gives you toxic stew.
Exactly. Once it enters the digestive tract, the amatoxin easily survives the harsh acids of the stomach and is rapidly absorbed through the intestines, traveling straight to the liver via the portal vein.
And once it's in the liver?
Inside the liver cells, it hunts for one specific target, an enzyme called RNA polymerase II. This enzyme is basically the fundamental engine of life.
Its job is to unzip human DNA and transcribe it into messenger RNA, right? Which the cell then uses as instructions to build new proteins.
Correct. The amatoxin physically binds to a moving part of this enzyme called the bridge helix. It acts like a microscopic steel wedge thrown into a delicate gear, permanently jamming the enzyme.
So if the cell can't transcribe DNA into mRNA, it can't build any new proteins, the entire biological assembly line just instantly halts.
Exactly. The cell stops functioning and begins to die. But the truly terrifying aspect of amatoxin poisoning is the delay.
The delay.
Yes. A person might eat a meal containing deadly galerina and feel perfectly fine for anywhere from 6 to 24 hours. The toxin is quietly shutting down their liver cells, but there are no immediate pain receptors firing.
They have no idea it's happening.
None. Eventually, the patient experiences violent, severe gastrointestinal distress, vomiting, and cramping. But then a cruel trick of physiology occurs. There is often a false recovery phase, sometimes called the honeymoon phase, lasting up to 2 days, where the patient feels significantly better.
Oh, no. So they think they just had a severe bout of food poisoning that has passed.
Right. But behind the scenes, the liver is quietly dissolving into necrosis. By the time the severe symptoms return, jaundice, bleeding, coma, the liver and kidney failure is often irreversible.
Wow.
Without an emergency organ transplant, the mortality rate is exceptionally high.
The stakes are just astronomical. You have a fascinating mushroom surviving in extreme conditions, perfectly mimicked by an absolute assassin. If they grow on the same log and look the same, how could a botanist or a cautious forager possibly tell them apart in the field?
It requires meticulous attention to a combination of subtle details. No single feature is enough. You really must observe them all. Let's start with how the caps dry out. Do you recall the hygrophonous nature of the wood tuft we discussed earlier?
Yeah, the wood tuft dries from the center, radiating outwards, leaving a dark, moisture-filled rim on the edge, creating a bullseye.
Gallerina marginata, however, typically exhibits the exact opposite drying pattern. It dries from the edge inwards. So a partially dried Gallerina will have a pale, dry outer rim and a dark, moisture-filled center.
Okay, that is a really helpful clue, but relying on the weather and the exact timing of evaporation seems a bit risky. What about the other senses? Smell and taste.
Ecologically possesses a mild, indistinct, generally pleasant mushroomy odor. Gallerina, on the other hand, strongly expresses what mycologists refer to as a farinaceous odor and taste.
Farinaceous. I've heard that described as smelling mealy, like a damp, musty basement or raw flour that has been sitting in a wet paper bag.
Musty raw flour is the classic description, yes. But you must also examine the stem. As we established, the wood tuft has a relatively smooth stipe that darkens from the base upward.
And the Gallerina.
The Gallerina possesses a silky fibrillose stipe. It is covered in tiny, silvery, silky threads, especially on the lower half, and it often has a more distinct, persistent ring from its veil.
Okay.
But the ultimate non-negotiable test is examining the spores.
Taking a spore print, cutting the cap off, laying it on a piece of white paper, and covering it with a glass overnight.
Correct. The millions of spores will drop and leave a colored footprint. Calignicola leaves a spore print that is distinctly rusty brown. The deadly Gallerina leaves a print that is a lighter cinnamon brown.
But even that color difference can be pretty subjective depending on the lighting in the room.
Exactly, which is why to achieve absolute certainty, you must use a microscope. Remember Scott Redhead's defining characteristic for the wood tuft.
Perfectly smooth spores with the flattened apical pore.
Gallerina spores, when viewed under high magnification, are rugulose. They are covered in tiny, distinct warts or wrinkles. If you are examining a sample and you see warty spores under the microscope, you immediately throw the entire basket into the trash.
The difference between a fascinating specimen and catastrophic liver failure comes down to smooth versus warty microscopic textures. It's harrowing. But as we hinted earlier, the wood tuft isn't just an innocent bystander worrying about poisoning humans. It is actively hunted itself.
Oh yes, the plot thickens.
Let's shift our focus to the threat that targets the wood tuft. I am utterly fascinated by the alien parasite.
This is undeniably one of the most bizarre and spectacular ecological interactions documented in the fungal kingdom. We are discussing an incredibly rare mycoparasitic fungus known as Squamanita fimbriata.
Mycoparasitic, meaning a fungus that explicitly preys on and eats other fungi.
Exactly.
We've been using the analogy of an alien body snatcher because this organism does not grow on soil. It does not possess the enzymes to break down conifer wood. It exclusively parasitizes the living fruiting bodies, the actual mushrooms of Quineromyces lignicola. It literally grows out of the host mushroom.
It does.
How does an organism orchestrate a biological hijacking like this?
The mechanics of the invasion are like something out of a science fiction horror film. The Squamanita exists as a stealthy mycelial network within the wood or the soil, basically just hunting for a host.
Okay.
When it detects the developing mycelial knots of the wood tuft, which are the dense bundles of tissue that are preparing to push up and become a mushroom, the parasite attacks.
Just ambushes it underground.
Right. The Squamanita hyphae physically invade the host tissue. As the wood tuft attempts to grow its stem and push up through the snow, the parasite systematically takes over the host's cellular scaffolding and developmental pathways.
It intercepts the biological blueprints.
Exactly. It completely aborts the development of the Calignicola cap. The brown, smooth cap of the wood tuft never actually forms. Instead, the parasite utilizes the hijacked stem as a nutrient pump, and a completely different mushroom erupts from the top, the Squamanita, which features a distinct, scaly, yellowish or purplish cap.
The body snatcher decapitates the host and grows its own head on the host's body. Is there any evidence left behind that the wood tuft was ever even there?
The only remaining evidence of the victim is found at the very base of the stem. There will be a swollen, deformed basal limb or a strange fibrillose annular zone.
Wow.
That distorted tissue at the bottom is the hijacked remnants of the host wood tuft. The parasite essentially uses the host's corpse as a launch pad.
That is beautifully grim. But there is a fascinating historical footnote in the literature about this specific parasite-host relationship that highlights how easily humans can misinterpret these complex ecosystems.
Yes, there is a major mycological correction on the record regarding this. For decades, the established scientific literature stated that the original type locality, the specific place in Norway where Squamonatus imbriata was first discovered, was parasitizing the common summer-fruiting wood tuft Cuneromyces mutabilis.
Okay.
But years later, advanced mycologists went back into the archives, re-examined the deformed basal limbs of those original Norwegian specimens, and realized the fundamental mistake had been made. The host was not mutabilis, it was our snowbank survivalist, Cuneromyces zinicola.
Why does that specific distinction matter so much? To a casual observer, mistaking one wood tuft for another seems like a pretty minor clerical error.
Because it completely changes our understanding of the parasite's evolution. It reveals a highly specialized evolutionary lock-and-key dependency.
How so?
The survival of this incredibly rare Squamanita parasite is 100% dependent on the extreme snowbank phenology of K. lignicola. The parasite does not emerge in the warm, easy days of autumn. It has evolved to wage its attack specifically in the freezing, melting snowpack conditions of late spring, because that is the exact narrow window when its specific host is vulnerable.
Oh, I see. So if the snowbank wood tuft fails to fruit one spring, the parasite just starves and goes extinct.
Absolutely. Their destinies and their biological clocks are completely, inextricably bound together in the alpine ice.
It is a stunning realization. We are looking at an entire, vicious, microscopic, ecosystem-acidic chemical warfare, biological antifreeze, lethal neurotoxins, and body-snatching parasites, all playing out on a single, rotting spruce log hidden under the snow.
It's an entire universe on one log.
Which brings us wonderfully to our final area exploration. With its ability to conquer the snow, obliterate toxic wood, navigate deadly doppelgangers, and host alien parasites, it makes perfect sense that human cultures, who have lived intimately alongside these boreal forests for millennia, have historically viewed wood tufts with a deep sense of reverence and utility.
It really is always illuminating to see how traditional ecological knowledge recognizes the value of these organisms long before we invent the tools to analyze them chemically.
The literature highlights a rich cultural significance that predates modern science. The sources look at Eastern Europe and Scandinavia. In Finland, the common name for the wood tuft is a direct acknowledgement of its extreme phenology.
Yes.
It is called kvetkantaisieni, which literally translates to the spring stump room. They recognize its unique timing centuries before we were running NMR spectroscopy.
Exactly. Indigenous and traditional forest cultures were exquisite observers of phenology. And in many of these Northern European and Slavic traditions, the wood tuft achieved a mythological status as a boundary fruit.
I find that phrase incredibly poetic. A boundary fruit. What exactly does that signify in the context of folklore?
It was viewed as a powerful physical symbol of transition and resurrection. Consider when and where it appears. It grows exclusively on dead, decaying matter, the rotting corpse of a massive tree.
Right.
Yet, it emerges right at the bleeding edge of the melting snow. It became a profound symbol of the forest, actively reclaiming the dead matter of winter, utilizing decay to fuel new life.
So it marked the literal physical boundary between the death and starvation of deep winter and the approaching vitality of spring.
Yes. For a foraging culture, finding that first massive golden-brown cluster of wood tufts aggressively pushing up through the freezing snow was the ultimate sign that the harsh winter was finally breaking.
And they didn't just revere them symbolically, right? They consumed them for health. The historical records indicate that wood tufts, including the spring varieties, were traditionally utilized in Eastern European broths, specifically to support immune health and recovery after the long winter.
They were heavily utilized.
Now, modern pharmacology often looks down on traditional folk remedies, but the chemical profiling we discussed earlier actually provides a mechanism for this, doesn't it?
It absolutely validates the traditional practice. When modern researchers isolated the secondary metabolites of the Cuneromyces genus, they didn't just find trathose antifreeze and sequestered heavy metals. They discovered a rich concentration of complex polysaccharides, specifically beta-glucans.
I've seen beta-glucans marketed in modern health supplements. What exactly are they doing at a cellular level inside the human immune system?
Beta-glucans are potent, naturally occurring immune modulators. When they are ingested, perhaps extracted into a hot broth, they interact with specific receptors in the human digestive tract, notably the dectin-1 receptors found on macrophages and natural killer cells.
Macrophages being the white blood cells that constantly patrol the body, engulfing and destroying bacteria and cellular debris.
Correct.
Yeah.
The beta-glucans bind to these cells and essentially act as a molecular training mechanism. They stimulate and prime the macrophages, putting them on high alert and increasing their phagocytic activity, their ability to consume pathogens.
Wow.
But crucially, they do this without triggering a massive systemic inflammatory response like a fever.
So the traditional Eastern European broths weren't just warm water. They were highly effective, low-grade chemical extractions of bioactive compounds that genuinely supported immune function.
Exactly.
That would be incredibly valuable to a population emerging from a long, nutritionally scarce winter.
It is a perfect example of folklore acting as applied generational chemistry.
Knowing what we know now, from 19th century visual taxonomy to 20th century electron microscopy to 21st century NMR spectroscopy, what is the next frontier? If you were handed a blank check grant to study Cuneromyces lignicola tomorrow, what are the researchers looking for next?
The undeniable future of this research lies in genomics and biotechnology. We have successfully mapped the metabolome. We know what chemicals the fungus produces. The critical next step is sequencing its entire genome to understand the exact genetic machinery of how it produces them. We need the source code.
Finding the specific genes responsible for these extreme adaptations. If we find them, how do we use them?
The applications are revolutionary. Imagine successfully isolating and sequencing the specific gene clusters that trigger the massive synthesis of the trehulose antifreeze during rapid temperature drops.
Okay, what could we do with that?
If we can understand that genetic trigger, we might be able to genetically modify critical human crops to survive sudden, devastating spring frosts.
Oh, wow.
But even more profoundly, it could revolutionize medical cryogenics. We could apply the fungus's vitrification technique to the cryogenic storage of human tissues and vital organs, preventing cellular ice damage during transport for transplants.
Taking a microscopic survival mechanism from a frozen subalpine log and using it to preserve a human heart on ice, that is incredible. And what about the lactase enzymes, the camlac that operates in highly acidic, toxic environments to obliterate conifer lignin?
The industrial potential for camlac is massive. Currently, the paper recycling and timber industries use harsh, toxic chemicals and immense amounts of heat to break down lignin and separate wood fibers.
It's a very dirty process.
Very dirty. If biotechnology companies could sequence the camlac genes and mass-produce this specific acid-stable enzyme, we could revolutionize the industry. We could use these enzymes to cleanly and biologically degrade lignin.
That would be huge for the environment.
Furthermore, because camlac thrives in toxic, acidic environments, it could be deployed as a highly efficient biocontrol agent to break down complex, toxic pollutants in the acidic runoff from mining operations. The genetic blueprint of this tiny mushroom holds the key to major industrial bioremediation.
That is just wild. Let's take a step back and recap the immense journey we have taken today in this deep dive. We began by staring at a tiny, unassuming brown mushroom shivering in the melting subalpine snow. We traced its chaotic 150-year history, starting as a misunderstood taxonomic enigma dumped into an overflowing botanical bucket, bouncing from genus to genus, until a half-micrometer, flattened escape hatch on its microscopic spores finally revealed its true evolutionary identity.
And then we unraveled its brilliant, brutal snowbank strategy. We saw how it deploys biological trefulose antifreeze to survive cellular rupture, completely avoiding seasonal competition. We examined its deployment of specialized, acid-loving camlac enzymes to weaponize the conifer tree's own toxic defenses, obliterating the dense lignin fortress.
We peered inside its chemical vault, discovering how it hoards toxic heavy metals like cadmium, permanently locking them within its cellular walls to prevent its own destruction. We navigated the terrifying reality of its deadly doppelganger, the amatoxin-load Galerina marginata that jams the engines of human liver cells.
And we witnessed the sci-fi horror of the body-snatching squamanita parasite, entirely dependent on the extreme timing of its host.
Right. And finally, we saw how this singular organism bridges the gap between ancient immune-boosting folklore and the cutting-edge future of genetic biotechnology.
It serves as a profound, humbling reminder that the most easily overlooked organisms in our ecosystems are often executing the most complex, highly evolved survival strategies on the planet.
It truly does. But before we wrap up this deep dive, I want to leave you, the listener, with one final lingering idea to ponder. We spent an hour discussing how modern scientists are mapping the internal chemistry of this fungus. But the application of that science is taking a truly mind-bending turn.
It really is.
The chemical fingerprint of Cuneromyces lignicula, specifically those amino acids in that protective trachelose, is incredibly stable over long periods. So stable, in fact, that researchers are now performing a revolutionary technique called historical metabolomics.
It is a brilliant intersection of history and biochemistry.
Scientists are not just foraging for new mushrooms in the snow. They are going deep into the botanical archives. They're opening up dusty museum drawers and taking microscopic samples of dried herbarium specimens of the wood tuft that were collected by botanists 50, 100, even 150 years ago.
And they are running those century-old tissues through the advanced NMR machines today.
Because the fungus absorbs and concentrates the exact chemical reality of its immediate environment, these dried specimens are not just dead tissue. They are highly accurate miniature biological time capsules.
Exactly.
I want you to imagine the sheer scale of what these tiny dried mushrooms sitting in dark museum drawers are currently revealing. By comparing the exact chemical makeup of a wood tuft picked in an untouched forest in 1872 to one picked during the height of the Industrial Revolution in 1950, to one picked from a melting snowbank today, scientists can read a century and a half of atmospheric history.
It's breathtaking.
They can track the exact rise and fall of atmospheric nitrogen deposition. They can measure the precise spikes in heavy metal pollution from long-closed, forgotten factories. They're using the preserved tissues of these undercover operatives to record the silent, shifting baseline of climate change in our oldest, most remote forests.
It is a flawless biochemical archive of human impact, permanently written into the cellular walls of a fungus.
So the next time you find yourself walking through a thawing forest as the spring snow begins to melt, and you happen to notice a cluster of tiny brown mushrooms shivering on a rotting log, do not just walk past them. Take a closer look. Because you might just be looking at the ultimate biological operative hiding in plain sight, hoarding our toxic secrets, and quietly recording the history of the world.